We're going to start with Michael COURTNEY, who's here all the way from Finland, from the Turku Bioscience Centre, who's going to talk about potential druggability of SYNGAP missense variants and how we might get there.
Okay, thank you very much, and thank you to SRF for organizing this great meeting and for giving me a chance to participate and present. Our lab is focusing on cell signaling related to neurological disease. So the aim is to identify druggable mechanisms, by which I mean some protein-mediated event that can be modified by a small molecule in a way that's hopefully beneficial for a disease. We work mainly with optical assays, and one thing about these is that they're usually very easy to automate and to put into a high throughput usage. And there are new developments coming all the time, so what we can do tomorrow may be much more powerful than what we can do today. So SYNGAP1 is a signaling protein we've been interested in for a long time, but we haven't really had a chance to really focus on it until recently with funding from Leon and Friends in Austria and SRF in Europe and US, that we've been able to really think about it from the disease perspective. So maybe I don't need to say too much for this slide, but the problems with missense really come about that we usually have really little idea what is actually wrong with the SYNGAP protein when there is a missense. And of course, we really need to know that in order to be able to figure out what therapeutic strategies we might take to try to improve it and not to cause harm. And another problem is that in many cases, there's only a single case known for a particular missense. And on the other hand, there are cases of particular missenses which are more common. There are multiple cases, and it seems like the symptom presentation is a little bit different, which means there's an impact to genetic background that kind of makes sense, because SYNGAP doesn't act alone. SYNGAP acts in concert with other proteins in the cell. And so this will complicate analysis, but I'll come back to that later. So again, for this slide, maybe I'll just point out that we're thinking repurposing would be a good starting point. But first, we really have to clarify the mechanisms of missense. And we have so many different missense that we potentially need to consider how are we going to manage this large number of missense and then think about repurposing on such a large diversity of SYNGAP variants. So hundreds of missense have already been described. We can simply go to ClinVar, for example, and we can find there's a categorization for each of them, all the way from pathogenic to benign or somewhere in between. And if we just plot this out according to the location of the missense along the SYNGAP protein, from N-terminus to C-terminus, we get a picture like this, which actually isn't so useful except to tell us that the vast majority of the categorizations are variants of uncertain significance, which is itself clearly a bit of a problem. If we do a bit of normalization to that data, we can see a bit more now. So we can see, I don't know if I can point, how do I point with this? Is that point? Oh, no, that's not going to work. Okay, so anyway, you can see that, for example, the pathogenic categorizations mainly around the GAP domain, which has a kind of relatively known function, and the C2 domain, which doesn't have such a very clear function, whereas conflicting interpretations are much more prevalent on the C-terminal region of the protein, which includes this DUF, domain of unknown function, and two domains, the coil domain and the PDZ binding motif, which do have fairly well understood functions. So what we'd like to do ideally is to understand each missense, which means we need to measure the function of each missense, and if there's something wrong with the functionality of a particular missense, ideally we'd find functional modifiers for each of them. But how do we approach this? So first thing is we need to figure out what kind of systems we should be using to measure function, and whether we can somehow miniaturize and automate the assays that we put together to measure function, so that we can now see whether small molecules will improve those functions. So I'd like to thank JR for a helpful discussion to kind of put a bit of a framework around this, so we can define the functionality of a missense in this kind of way using genetic terminology. Either we have a normal function, there's not actually anything wrong with the missense, that's quite possible, so that's an isomorph, or there could be too high activity, too low activity, no activity at all. On the other hand, a missense potentially could interfere with the normal function of the wild-type allele, so we have a dominant negative function, or we could even have a completely novel function at this point we perhaps can't even imagine. And the question is, how are we going to find these? So, and the problem is, of course, in each case, the answer will affect the potential therapeutic approach. So when we talk about activity here, it's not necessarily one measure. We've heard a lot about the different functions and non-synaptic functions of SYNGAP already today. I'm just focusing on activities that have been described relevant particularly to the synaptic presence of SYNGAP in differentiated neurons. And there's quite a few of them already here. So the question is, how do we measure all these, and what kind of throughput can we get? We have, for example, in our lab, a bunch of automated microscopes, plate handlers, and liquid handling devices. So what we're trying to do is set up assays that will run on these kind of systems, so that we know that we'll be able to run at a reasonable throughput once we have the assay set up and giving a robust readout. And then when it comes to model systems, we work with purified proteins or model lab cell lines like 293 cells, or primary cultured neurons, usually from rodent, or sometimes human neurons. And I think these are the key models that we can use. I've put up a little chart here of which models appropriate for which kind of readout, depending on the readout. This is somewhat debatable, but this is our view here. So, in fact, the human neuron system is, in a way, perhaps the best one, but unfortunately there are a number of issues that make it quite difficult to use, at least in a high throughput manner. So we tend to use some of the others, mainly the rodent neurons expressing SYNGAP from an AAV gene transfer vector. Okay, so I'd first like to talk about protein expression. So SYNGAP1 is haploinsufficient, so it's a reasonable hypothesis that a missense could cause a disease simply by preventing the protein from being expressed to a sufficiently high level. And sometimes there's a tendency to assume that from a starting point, and yeah, that's reasonable, but maybe there's not a lot of experimental data on the protein to really demonstrate this. But a couple of years ago, there was a very comprehensive study from Kurt Hass's lab with 57 different SYNGAP variants. You can see the disease-associated ones in the blue bars here in this particular part of one of the figures that I've pulled out. So this figure part is about expression levels, and you can actually see that the expression's pretty normal across the board from one end to the other, except for a couple of extremes with a low expression or high expression. So it's not obvious from this kind of analysis that there's something intrinsically wrong with the missense that prevents it from being expressed. On the other hand, for convenience, this was done in a standard lab cell line, a 293 cell, which is not a neuron, doesn't have synapses, doesn't have synaptic proteins that SYNGAP associates with. So you could ask the question, would there be a different interpretation if we would ask this question in neurons? So I'll come to that in a couple of slides. But before I get there, there's another way of thinking about protein expression and stability. That is, how stable is the structure of SYNGAP? If you put in a point mutation, which is what's happening in a missense, then perhaps the protein is not able to fold properly. So we can evaluate that directly with a thermal shift assay. We throw in some energy in the form of heat to the protein, and then we see how much it can tolerate before it starts to denature. This is a fairly standard assay, and it can be done in high throughput. We were testing a couple of methods. Here's some results from one of them we started with. This is perhaps a bit noisier than the new one we're starting, but we can see in the bottom graph the wild type has a higher stability, and all of the handful of patient variants we're looking at the moment seem to have a slightly lower stability that's quite measurable in this assay. So this is definitely something that we can put a number on, and we could potentially run a screen with these kind of this kind of approach. Okay, now I was asking, well, how does SYNGAP look in neurons? So we can express with AAV at reasonable expression levels wild-type SYNGAP, and we see in this figure at the top on the left-hand side the distribution of SYNGAP. We see these puncta, presumed synaptic localization, as it should be. And then if we see the two images on the right-hand side, then there is the distribution with a couple of missense variants. And we can see it looks pretty different, but if you look closely, there are also puncta in both of these cases. So the question is, what is really the difference here? So we can quantify in an automated fashion the intensity or the average intensity of a puncture, and we see this in the bar chart at the bottom. So yes, there's a difference among 3 missense compared with wild type. Or we can actually make an average of the intensity of all of the puncture that we can measure, and that takes into account the number of puncture, the size of the puncture, and the intensity of the puncture, and you can see a bigger difference. Or we can apply a kind of unsupervised clustering approach here on the bottom right, and it's good to see that it's actually quite easy to automate the analysis and easily distinguish wild types in pink and purple here compared with the 3 missense variants we're looking at here, which all seem to cluster together. So that's kind of interesting as well. 3 different missense variants all have similar properties in terms of the features we can measure from these puncta. So another thing that SYNGAP's supposed to do is to respond to neuronal stimulation. It's supposed to leave the synapse in response to neuronal activity. And here we have, it's actually supposed to be a movie, but it's fine to just look at it as a still here, because what we can see in magenta is the initial localization of SYNGAP1. You can see it along the sides of a dendrite here, and the dendrite's highlighted in cyan, because cyan is actually the image of the final location of SYNGAP after the stimulation. So you can immediately see that there's a big difference between the initial and final location of SYNGAP. And the nice thing about this assay is it's been induced in an optogenetic manner, which means there's no liquid handling on the stage during the assay. This is, again, pretty easy to automate. So I think this is a method that can also be used for screening to measure that function. Okay, so SYNGAP is known to be able to regulate the RAS-ERK signaling pathway, and in this case we're using liquid handling to induce neuronal synaptic activity. You can see that in the middle figure. There's a little pipetter on the stage of the microscope, and on the top right you can see in pink when we knock down SYNGAP1 we see a increased activity of the ERK pathway, which is what we expect, and then we can, in the bottom right, we can reintroduce human SYNGAP wild type. Or we could, in principle, introduce a missense variant, and so we can quantify whether the missense is able to impact the ERK pathway as much as the wild type or not. This is clearly, you can see from the error bars, it's a noisy assay. It could be used for validation, but I think it wouldn't be so good for screening. I'm probably running low on time, because 5 minutes. Okay, thanks. So I wanted to highlight the C-terminus of the protein. This is a very nice study from Mingxi Zhang's lab that's demonstrated by crystallography and chromatography the formation of a trimer caused by the C-terminus. And this is a very elegant study, but in terms of functionality of the full-length protein and the effect of missense, only the C-terminal fragment is present in this measurement shown here. So we would like to know whether we can actually, first of all, put this is also in cell-free systems. So first of all, can we put it in cells and make a measurement of the interaction in cells, and we seem to be able to do that with a resonance energy transfer method. We can also look at localization of the protein with PSD95. But again, in both of these cases, it's only the fragment. Even though it's in cells, it's only fragment. So we cannot ask questions about the missense at the N-terminus of the protein affecting this interaction. So we are actively looking into methods. There are methods available to quantify protein interaction and protein oligomerization in intact cells. It could be applied to neurons. It can be applied in an imaging format, so we can look at synapses and extrasynaptic regions. And this is the way we're going with this, to try to bring this forwards. When we come to neomorphic activities, it means that we actually don't even know what we're looking for, so we have to look at an omic and a proteomic-wide way. And so we've been setting up proximity proteomics. Lily in the lab has just got back some setup data to check that the method is going to work. And if it all looks good, we can then move to the SYNGAP case. This is being assisted by a neighboring proteomics facility, which is really expert in these methods. So we have high hopes that this will give us information about whether missense is interacting with new proteins that normal wild-type SYNGAP is not. Okay, but in summary, what we're trying to do is to generate screen-compatible readouts of missense function and develop it as a proof of concept with a few missense variants, and then apply this to a wider range of missense so that we can then group the missense according to their similar properties. For example, you've already seen the C2 domain missense. The three of them we've been looking at actually are quite similar in terms of the puncture features, grouping together. We could then select one member of that group for a repurposing screen, and then once we have hits, we can then go back to other members of the group. So that's our strategy. I did mention this impact of genetic background, and this is a bit problematic. What I've been talking about in the assays so far has been to examine what is wrong with a missense SYNGAP by just putting that missense SYNGAP into the same kind of neuronal environment. But if there's an interaction between the genetic background and the SYNGAP missense, then we have a problem. It won't be sufficient just to look at the missense. We have to have the patient background as well. And that's where I think the neuronally differentiated patient cells are really important. And in that case, we have to be able to extend our assays into these human neurons derived from patients. And there, I think the proteomic methods will be particularly important as well. Okay, with that, I'd like to thank the people in my lab. I mentioned Lily and Afra and Barbara in the slides, but also other members of the lab are contributing to this project. Pekka's team you'll hear from in the next talk. It's been very helpful as well. And the proteomics team and IT people and contributions from Gavin, Anuburu, Winfried, and Matthias have been useful for us as well. And I'd like to thank all the funders who've helped us put this kind of approach together, and particularly SRF, of course, for helpful discussions as well as the funding and fundraising. Thank you.